Hello everyone, welcome to this new module on the course Mathematical Aspects of Biomedical Electronic System Design. This is a TA session. My name is Anil. I am a TA for this course. Professor Chandramani Singh and Professor Hardik J. Pandya has already covered the several fundamentals of the mathematical aspects of how to design biomedical systems. In this session, we will take a deep dive into the introduction to electrical equivalent circuit models for different biological systems. So, before we start off, we need to understand how tissues are organized in the biological system. So, this is a very interesting graphic which we have borrowed from the famous Max Planck Institute of Molecular Cell Biology and Genetics. So, what is tissue organization? That is what we have shown here. What is tissue organization? For that matter, what is a tissue? And how do tissues make up biological systems? So, before we delve into electrical equivalent models of biological systems, we need to understand how these systems come together, how they assemble and form organisms. So, let us go back from the bigger abstraction to the smallest unit of the whole organization. So, all of us know that we are all organisms. Man is an organism, human being is an organism. This mice and rats shown here, these are all organisms. They are capable of independent existence, and depending on the level of development of their brain function, some members of this organism are also capable of making decisions of their own. So, these are aspects where we identify certain entities as organisms. Now, coming down, so organism, like let us say human being is composed of different organs. This is basic high school biology. All of us have learnt about this basic high school biology. So, each organism is composed of different organs. So, organs and organ systems actually. So, in between organism and organs you have actually organ systems. All of you might have studied. So, what is organ system? Let us say our circulatory system is an organ system. The nervous system is an organ system. So, what is an organ system? Organ system is a collection of organs within an organism that can perform a set of functions. Let us say circulatory system, if you take, it can perform pumping of blood, and oxygenated blood is pumped through the body, then the different parts of the body use this oxygen, and then it is circled back to the circulatory system where the deoxygenated blood is again revitalized with oxygen and pushed back into the body. So, that is the circulatory system. So, like this we have different organ systems. Now, what are these organ systems made of? Yes, these organ systems are made of organs. Let us say if we again take circulatory system, it is made up of heart is an organ. If you take pulmonary system, lung is a very important part of it. If you take nervous system, brain is a part of it. So, these are all organs, individual organs. So, that is the next level of abstraction below the organ system. So, first we have organism, then we have organ system, then we have an organ. Okay. Now, what are these organs composed of? These organs are composed of a specific structured arrangement of cells along with blood vessels which supply nutrients to the cell. So, those are the lobules and tissues. Okay. So, let us say I make a very simple abstraction of this organ. Let us say I am taking liver as an organ, okay, and I am cutting a part of it. Let us assume that this is liver, and I am cutting a part of this organ here. And if we try to see it from a cross section point of view, what we see is an organization of cells which are the basic units of life. Cells arranged in either a structured order or a non-structured order. Depending on the organ, they may or may not have a structured organization. And then between them you have actually something called as matrix which holds these cells together. Okay. So, this organization is called tissue or tissue architecture. Now, just these cells, usually these are called epithelial cells. Epithelial cells, there are endothelial cells, different types of cells are there which come together to form tissues. Now these cells alone cannot survive, they need nutrients to survive, right? Who brings the nutrients? The nutrients are brought together by vasculature or the blood supply into the tissues. Okay, so it's a very complex arrangement, it's brought together by blood supply into the tissues. These are also tissues actually. These bloods are also blood vessels are also made up of specific types of cells called endothelial cells. I might be going into this, you just consider it as general awareness so that we understand the next part of the course. So this is just a general understanding about how organs are assembled by tissues and lobules. Okay. So cells themselves in a structured arrangement cannot exist on their own, they need to be supplied by nutrients, and those nutrients are supplied by these vessels like blood vessels, lymphatic vessels, etc. Okay. So now we have covered organism, we have covered organ system, we have covered organ, we have reasonably covered lobules, we have covered tissue. Now as I said, how do these tissues come to exist? These tissues come to exist when cells come together. Cell, I think all of you are aware. So cells are kind of like the individual functional unit of a living being, individual independent functional unit of a living being. So cells can come together, arrange in a particular form and form tissues, as we said before, right. So the way these cells come together and the kind of function that these cells perform will dictate what kind of tissue this is. So let us say we take muscle tissue. Here actually the cells are not like this square shaped, they are actually more spindle shaped. These are called muscle fiber cells. They are shaped like this, evolutionarily they have come to attain this shape so that they can expand and contract to perform the muscular functions. So that is why they have this striated, it's called striated epithelium. There are different types of muscle tissues. Just for example I am telling you. For another example is, let us say we take brain. Brain has lot of different types of cells, but one major cell which you all might be aware of is the neuron. Neuron has a different structure like this, with nucleus and these dendrites. These are used for conveying the electrical signals to the next cell. So these are neurons. So as I said, the cells do not necessarily have one particular shape, the shape is dictated by function. There is an important dogma in biology called form dictates function. So the form of this neuron dictates its function, which is to transmit electrical signals for brain function. The form of this muscle cells dictate its function so that it can expand and contract and perform muscular functions. So that is what cells, that is how cells come together to form tissues, and the shape of cells will be different for different types of tissues, and this change in shape, size and organization will define what is the end function of such tissues. Okay. So now we have covered that cell as an abstraction, cells as an abstraction come together and form tissues. Now what is inside these cells? That is the next part, that is a subcellular. So all of you, this is basic, we will only go into the basic details of 10th standard biology. We all know that cell is there, cell has a nucleus inside, okay, cell has a cell membrane, okay, and there are other parts of the cell called mitochondria, then Golgi bodies. The different organelles are there. These individual units inside the cell are called organelles. So nucleus is often called brain of the cell, right, you would have all learnt in 10th standard. Then there are other small organelles in the cell which is beyond the scope of this course. So what you need to understand here is that this cell membrane defines the structure of the cell. It's like, so in all your dimensions, in all the drawings you see cell as a 2D or oblong structure, right, but actually what is the cell? Cell is actually a 3D ball. It's actually a 3D ball, which is, it's like you can consider it like a droplet of water that is found on top of this lotus leaves. in a hydrophobic surface, if you imagine water, many of these cells are shaped like that. It's a 3D 3D what you call a 3D volume with this, which is optimized for the surface tension, and it will be circular or a 3D circular volume in shape. So, within this the nucleus is contained and you have the cell membrane. Okay. But the inside part of the cell is called the cytoplasm. Okay. Inside part of the cell is called cytoplasm, and for our important our understanding, the main thing to note about cytoplasm is that it is mostly consisted of water and ions. Different types of ions are the major constituents of cytoplasm along with the other organelles. Okay. So, the nucleus is very interesting. Nucleus is what contain is important because it contains the DNA, the deoxyribonucleic acid, which is like kind of like the software program for our overall functioning. So, nucleus hosts the DNA through chromosomes. All of you might have heard about the XY chromosomes, 22 pairs and all, right. These are all basic knowledge. And the cell membrane limits the shape of the cell and makes it stick together as a single entity. So, this is a basic structure of the cell. This is important because this will define how we understand the electrical equivalent model of the cell. Okay. So, I think this is clear to you. So, this is the structure of the cell. So, what are the major components that are needed to be understood in order to electrically model a cell are these: 1, cell membrane, okay, which is the outer membrane. It is usually consisted of a what is called as a lipid bilayer. A lipid bilayer, lipid is basically fat. It is a fat bilayer. If you zoom in, how it looks like is we have two walls, right, 2 layers of the membrane. There are two sets of lipids here. This organization is there because inside this it is a hydrophobic surface. That means it is afraid of water. Because this inside is hydrophobic and these parts are hydrophilic, it helps the cell to get its structure, because the cell attains a volume of minimum surface tension, and just like water droplet is forms on top of a lotus leaf, but depending on the surface and kind of organization that is there. So, cell membrane is mostly consisted of a lipid bilayer. Okay. Please note this. This is really important when we model the electrical equivalent circuit. Okay. So, what I told, cell membrane is consisting of lipid bilayer. Consists of lipid bilayer. Okay. So that is basically like there are 2 layers and in between there is a void kind of situation. Okay. Just keep this in mind. It will become interesting when I tell you what this translates to electrically. Okay, this is lipid bilayer. The next important thing is obviously the cytoplasm, or sometimes called the cytosol. Okay, this is the intracellular space, the space inside the cell. This is mostly consisted of ions and water. Okay, I am just telling the major components of it. Okay, this is required for our electrical modeling. Then we have the nucleus. Okay. Another so the nucleus, as I told, consists again kind of like with ions and water, but it also contains the genetic material or the DNA in the form of chromosomes. Okay. Now, the nucleus is interesting because nucleus is again like a cell. Nucleus by itself also has a nuclear membrane, and the portion inside the nucleus is again called nucleoplasm. Like cytoplasm, it is called nucleoplasm. I think many of you might have read about this in your high school, but I am telling this again because these are all contribute to how we model it electrically. Okay. So, this is nucleoplasm. Again we have nuclear membrane, which is again a lipid bilayer. Only thing is the nucleoplasm is very densely packed with the genetic material. The chromosomes, the 22 pair of chromosomes are densely packed here. Why I am telling densely packed is because our DNA material, it's just it's a very interesting fact which I wanted to bring to your attention. Our DNA material, okay, consists of 3 billion 3 into 10 raised to 9 base pairs of nucleic acids, which that is basically A, T, G, C pairs. You can read up about it. These are nucleic acids. This is what constitutes our DNA. Okay, this if you try to linearly put this 3 into 10 raised to 9 base pairs in a linear fashion, it will I do not know exactly, but it is said that it can go around the earth 2 times, I am not sure. So, it is very long. So, to contain it within the very small micron or even nano dimensions of nucleus, it gets heavily condensed. So, it is a very dense layer. Okay, this the genetic material is very densely condensed inside the nucleoplasm. Okay. So, that is the thing, and the nuclear membrane is much smaller as compared to the cell membrane. As you can see, if I draw the cell on top of this, the nucleus will be very small portion of the whole cell membrane. So, the nuclear membrane, if you go for a first level simplification, nuclear membrane is almost negligible. Okay. If just keep this in mind, it will be important when we electrically model it. Okay. So, these are the different constituents of the cell which are important to us from a biomedical system mathematical modeling point of view. So, basically it is cell membrane, cytoplasm, organelles do not contribute much to the electrical properties, I will come to that soon. Then nucleus again consists of nucleoplasm and nuclear membrane, and obviously the nucleoplasm consists of the genetic materials. So, this is the subcellular description of the cells. Okay. Now, within the subcellular space there are lot of proteins. Okay. Proteins are can be considered as how do we say, it's this they are an assembly of nucleic acid molecules or different types of molecules, and they, depending on their shape, they are able to perform a particular function. I think that much only we should cover here, because if we go into detail, it is an entire field in itself. Only thing is, the DNA that is inside the nucleus actually determines what all proteins get made, and these proteins that are made reside in the cytoplasm. So, a copy of the DNA, this is called the central dogma of molecular biology. Okay, it is a defining principle of entire field of biology, where the DNA, a copy of the DNA is made as RNA, which is ribonucleic acid, which is then converted to proteins. Okay. So, it is just like you having one master copy of your question paper, and then you make Xerox copies of the question paper, and these Xerox copies are distributed to students, and the students who know the correct answer to the questions write the answers, correct answers. These correct answers are the proteins. Each question paper given to the student is RNA, and the original question paper is the DNA, and it is very important that students do not make any changes to the original question paper, then the whole chain gets disrupted. That is why in the cell, first a copy of the DNA is made called RNA, and using the RNA we make proteins, so that the fidelity or the faithfulness of the DNA is not compromised. Okay. So, that is how proteins are made. Now, this was just a brief deviation from our core area, but these are interesting points. So, I wanted to cover that. Okay. So, this is a central aspect of biology, and that is how these molecular species come into existence. But to be honest, for our consideration, the proteins do not contribute much to the electrical equivalent model of the cell. So, we will not go into details of it. Okay. So, for us the main thing is, again I will repeat, I will keep repeating it because this is very important: cell membrane, CM, cytoplasm and nucleus. You need to remember only these. Nucleus again consists of nucleoplasm and nuclear membrane. Okay. Just consider these three things for now. Cells again then come together to form tissues. Tissues along with vasculature or lobules come together to form organs. Organs come together to form organ systems, and organ systems come to form independent self-aware organisms. That is how the whole life comes into existence. Okay. So, this is a brief introduction to overall tissue organization. I hope all of you are clear. If you have doubts, you can always post questions in the forum. Now we will go into the main aspect of this lecture, which is, so we wanted to make sure that you understand the whole beauty of biology, so that you can appreciate the electrical model also, and it does not become like a rote learning or by hearting as such, because you understand all these things automatically. If somebody asks you, you can automatically by default intuitively come up with the electrical equivalent circuit of any type of cell that is given to you. That is why we went into detail about tissue organization and what constitutes a cell, what constitutes the subcellular components of a cell, etc. Okay. Now let us get into the interesting stuff. Now what is, so now we will see the equivalent circuit model of a single cell. Okay. Let us start with the basics. Once we know the single cell, we can always bring the cells together and form tissues, bring the tissues together and form organs. Now, so this equivalent circuit model has been in place from a long time. I think even from 1960s people used to have the idea of an equivalent circuit model, but this kind of model it was very in detail explained in this paper by Morimoto et al. In 1990. This is often called the cold model. Okay, you can read it up. There might be minor modifications. We will take a separate lecture where we go through each of the different types of models that are there in literature, so that you have a good understanding about different models. But now let us not think about which scientist did what and all. Let us just try to understand what is a model. Okay. Now this is the cell as we have described. So this is the cell membrane. Okay. Like just now we covered. This is the cytoplasm. This is the nucleus. Okay, this is clear. Now what did I tell? I told you that cell membrane is a lipid bilayer, right? Okay, so that is why it is two layers are here. Now how was a lipid bilayer? I will zoom in. Lipid bilayer is like two layers of fat. If any biologist hears how I am explaining it, they might scold me, but because this is an electrical engineering course, I am simplifying it. So lipid bilayer is like two layers of fat separated by a void. Okay, this void becomes hydrophobic and this edges become hydrophilic. Okay, now what does this look like to you? What does this look like to you? Yes, this looks like a capacitor, right? We have two parallel plates separated with a dielectric membrane. That is our capacitor, right? So this is like capacitor only. Okay, but then there is a small catch here. This lipid bilayer is not always continuous. Okay, in some locations there might be openings. That opening, why is that opening there? It is interesting. Why is an opening there in the lipid bilayer? Obviously it should be there, because how else will the cell take in things that are outside the cell so that it can survive? So it needs to have a ion balance. So there is a sodium potassium balance equation that is done. Sodium ions come in, potassium ions go out, and some nutrients come in. So these things happen. The cell cannot exist on its own, it needs nutrients from outside, right? So these small ion channels, these are called ion channels, allow some things to come in and go out through the cell membrane at specific locations in the overall membrane. So if I draw the membrane here, let us say I draw the membrane here as a big entity. There are two layers, right? If I draw the membrane here, there will be random openings here and there, not many. Okay, so what would these random openings be then? They will be like a resistance, right? They will be like a low resistance path for an electric current to come through, because in all the other places it is just like a capacitance. In some places there is a direct path, but it is not a direct path like a short, but it is a sufficiently resistive path. That is why you can model the cell membrane as a parallel combination of a resistor and a capacitor. You can name it as CM and RM, membrane capacitance and membrane resistance. That is why CM and RM. That is what is shown here. So that part is done. You have understood. So that is the electrical equivalent model of the cell membrane of the cell. Okay. Now that is done. Now what did we see next? What did we see next is that we have the cytoplasm. Here as you can see, the cytoplasm is modeled as a simple resistor. Now you will obviously ask me, how can that be there? Cytoplasm has lot of organelles, there are a lot of proteins running around the cytoplasm. Okay, there are the mitochondria, there is Golgi bodies, lot of things that you have learned. there are some things like the suicide bags of the cell, lysosomes or liposomes or something we used to study right in school. There are a lot of things, it's a site of intense activity, there are a lot of things happening in the cytoplasm. Then how can you model the cytoplasm as just a resistance? How can that be happening? Yeah, this is where our basic circuit understanding needs to come in. What did I tell you? The cytoplasm is mostly consists of the liquid part of the cytoplasm, mostly consists of ions, water, basically water in its ionic state, H plus and OH minus, and other ions like sodium ion, potassium ion, lot of ions will be there. Okay. Now imagine a circuit where you have a low resistance, low value resistance, let us say 1 kilo ohm resistor connected in parallel with very high resistances like 200 kilo ohm, 300 kilo ohm, etc. Okay. Now what is the effective resistance? Let us say we are just simplify it, we are just connecting it with a very high resistance and a low resistance. Okay. Now here this low resistance is the ions, because ions as you know are quite conductive, right. So they will have very less resistance. Let us say this 200 kilo ohm is from the organelles, because they are like, they contain this like this lipids and proteins and all, they are heavy molecules and they will have high resistance. Now what will be the effective resistance of this parallel combination? It will be 1 into 200 by 201, right, R1 R2 by R1 plus R2, simple first year engineering topic. So R1 R2 by R1 plus R2 kilo ohm. So what this will be? This will be 200 kilo ohm by 201 kilo ohm. Basically it will be less than 1 kilo ohm, right. So what that means is that the effective electrical nature of the cytoplasm is dictated by the heavily conducting ions inside the cytoplasm. That is why no matter what all things are there inside, including the nucleus, it is not going to affect the effective electrical property of the cytoplasm, because it is overpowered, or it is majorly dominated by the highly conducting ions in the cytoplasm. That is why it is modeled as a resistor. This Ri means ionic resistance, you can tell, intracellular resistance, you can call it in different ways. So this is R intracellular, inside the cell. Okay. So this is clear, right. These are very important conceptual understanding. This is not very in detail covered in any of the curriculum, actual physical curriculum in the country. So these are things that we have come to understand, part of our research that we are sharing with you. I hope this is useful and you can appreciate the importance of these things, and more than appreciating it, I think you can understand the beauty of it, and the how interesting this whole thing is, how biology, our basic unit of life, is getting modeled as abstract resistances and capacitors, which we only play around with in breadboards, right. So that is the reason why the cell membrane, and the cell membrane is modeled as the capacitor and the resistor parallel, and you understood the reason why the cytoplasm is modeled as a resistance, right. Now for simplicity, because if you are, let us say you are taking a single cell, it is a very hypothetical situation, it is very difficult to apply current to a single cell. Though there are now technological advancements where we can do that, but let us say we are applying current through a single cell, current has to enter from one point and exit from one point, right. So this cell membrane is can be modeled as two such interfaces of resistors and capacitors. That is a simple thing. So two such interfaces of resistors and capacitors. So that is what we are seeing here, these two, and it goes through the cytoplasm, which is Ri. Note here that the nucleus is not at all coming into picture, but why have we drawn the nucleus? I will come to that. Now the cell does not exist on its own. I told you many times, cell cannot exist on its own, it has to structurally arrange itself around something called as a matrix. Okay. Now this matrix is usually consists of many different materials, but mostly it is called extracellular matrix, is mostly consists of fibers, you can say, like collagen fiber it is called, but you can just think it as fiber. So these are like non-living entities, and they are like resistances only, like you take a very high resistance resistor and you are just connecting it, it is like fiber, just fibers. So that is why this collagen will come. Let us say now I take I remove this part of the tissue where I take one cell along with its associated matrix and keep it aside. It is a very hypothetical explanation. So I take a cell and I take its associated fiber, okay, this is cell, and then I apply current through it. What will happen? So this current will go through this resistance in parallel with the effective model of the cell, that is this part. So, effective model of the cell and the RE, what is RE? RE is extracellular matrix, resistance of the extracellular matrix. So, that is why RE is there. So, RE will come in parallel, because you can see, right, RE is coming in parallel with the effective model of the cell. That will define how a cell and its matrix responds to the electric current. Okay. So, this is the basic equivalent circuit of the cell. Now, you have understood, right? Then you are like obviously asking why have we drawn the nucleus? It is not even participating in it. One thing is sense of completeness. Without the nucleus, the cell itself does not have any purpose in existence, because the whole genetic material is inside, right? But for our electrical modeling, it is not contributing. But there are conditions like in cancer and other diseases. What happens is the nucleus becomes extremely large and also becomes multinucleated, or multiple, more than one nucleus comes in conditions of disease. What is this course about? This course is about designing biomedical systems. Why are biomedical systems designed? Biomedical systems are designed to diagnose diseases, to help in surgery, for different biomedical applications, right? So, in disease condition, what happens? I will simply tell you, I will not go into detail. What happens? This nucleus, no, this small nucleus might become this big, and it might become multinucleated like this, and along with the membrane. In this condition, it may not be a good assumption to omit the contribution of nucleus. We do not know, it may not be a good assumption. That is why the nucleus is there, that we have to go in a case by case basis and see. So, that is one thing, because the nucleus also has nucleoplasm, right, which is again quite conducting, but it is not as conducting as cytoplasm, because it is a dense region with lot of genetic material. So, this is very complex modeling. This is the thing of research. These are the things that people doing mathematical modeling research in biomedical systems do. I am just introducing you to this flavor, get a flavor of all these things. So, that is why the nucleus is still shown, because there are conditions like in cancer where the nucleus might become bigger and contribute to the actual effective equivalent circuit of the cell. Okay. So, I think this is very clear for you. As I described earlier, only thing that you need to think about is the cell membrane, the cytoplasm, and the extracellular matrix. Nucleus to some extent. So, cell membrane is a parallel combination of a resistor and capacitor, the Cm and Rm, membrane capacitance and membrane resistance. You understood why this resistance is coming, right? And then the intracellular space consists of Ri, the intracellular resistance, and you understood why it is only a resistance, why it is modeled as only a resistance. You understood why there are two pairs of resistor capacitor parallel combinations for the membrane, and you understood what is the extracellular matrix and why it is modeled as a resistor, right. This is the basic quite exhaustive way of looking at the equivalent circuit of a single cell. Okay. Actually, this is all what you need to know. Once you know this, you can always build on this and come up with new circuits where different cells come together. Okay. So, we will see some examples. So, again this is just a repetition of what we have told. Lipids are insulators, capacitance and resistance, and then cytosol are ions and organelles conducting. Extracellular space is water, ion, and fibers. Extracellular space also has water, okay, by the way. So, it is also, that is why it is also resistive. Now, this is like we are just seeing how can we see, how can we model the cell as resistor capacitor combinations, right. Mathematically, how can we have a equation equivalent circuit of a with a physical significance. Now, we only modeled it, but what are we applying across this? Are we applying a DC voltage? Are we applying an AC voltage? What if we apply a DC voltage? What if we apply high frequency AC voltage? What if we apply a low frequency AC voltage? How does it change? That we will see next slide. Now, once you have understood the basic property of the cell which we just covered, these things should come to you automatically. Now, I will help you out, but by the moment I explain, it will be very obvious to you. Okay. Now, what did I tell you? The cell membrane is capacitive, right? C. So, what is the nature of a capacitor? The capacitor, what is the impedance of capacitor? Let us say 1 by C omega, right, that is the impedance of capacitor, right. Omega is what? The frequency. So, what that means is when frequency is 0, the capacitor offers infinite impedance. Basically, capacitor blocks DC, correct. and very high frequency capacitor almost becomes close to 1 by infinity, which is roughly 0, it becomes very small value, right. So, what that means is that at low frequencies, let us say I take the spectrum, this is very high frequency, this is low frequency, this is DC 0 hertz. What that means is at very high frequencies, let us say this is the impedance of the capacitor, at very high frequency the capacitive impedance is low, at very low frequencies the capacitive impedance is very high, right. That means it is very difficult for the current to pass through at lower frequencies because of this 1 by c omega relation, which is very obvious, I think all of you understand. So, because of that what happens, that means at the moment of first instance when the cell sees the alternating current, if the alternating current is of a very low frequency, let us say 20 hertz, which is very low frequency, the membrane itself offers very high amounts of resistance or impedance to the flow of current, in the effect that the current skips the cell and goes around the lower resistance path, because this is just a resistance, right, outside the cell, it decides to go around the cell at lower frequencies. But in higher frequencies what happens, the capacitor, the membrane, the capacitive lipid bilayer has very the capacitance of the membrane has very low impedance, right. So, it will allow, let us say instead of 20 hertz it is 20 megahertz, very high frequency, the current can pass through because it is a low resistance path now, low impedance path now. This is the frequency dependent behavior of cells that are clustered together. That is what is shown here. This is from a paper from Journal of Visualization, we have adapted it from there so that you can understand it. So, this is what is shown here. See, high frequency component, the current is able to pass through the cells directly. The low frequency component tries to avoid the cells like this and tries to pass through the matrix. This has very important implications of how a tissue, what will this be? These are all matrices here, right, as we told, this is all matrix around the cell, full matrix. So, this matrix and cells coming together is what constitutes tissue, right. So, this behavior defines how the tissue responds to low frequency excitation and high frequency excitation. That is how the impedance response changes, and depending on different kind of tissues, the way the cells are arranged, the percentage of matrix and cells in the tissue, some tissues might have lot of matrix and very less number of cells, they will behave differently. Some tissues may have very less matrix, mostly cells only they will be there, they will behave differently. So, you will be able, when somebody tells you, okay, this is the structure of my tissue, now tell me what will be its equivalent circuit model and how will it respond to an AC signal of so and so frequency. Now, you should be able to intuitively tell them that how it will respond. You may not be able to tell exact values of what impedance will be there at what frequency, but you will always be able to tell, okay, it will have a very high resistance to flow of current at this frequency, but at this frequency it will be low. Those frequencies you can get by the equivalent circuit parameters, these RM, CM, RICA parameters of the tissue. Okay. So, this is the frequency dependent behavior of cell clusters. Okay. Now, one last thing is, let us say this is 3D, we are seeing a 3D volume of cells, right, arranged in a tissue. Cells are there like this. This is a 3D volume of cells and you are applying current, correct. So, this is what we saw, and we saw how low frequency will bypass it and high frequency will go through the tissue and all, right. Now, let us say imagine that we have a plate where cells are grown, 2D plate, and we have electrodes below it, and it is a single mono layer of cells that are growing. It is like a surface where one layer of cell only is growing like this, nucleus, and these are electrodes. How do we model that? That is what is modeled in this model. This is from Professor Pandya's one published paper. We can share the link to that paper, you can go check it out. So, basically what that means is this tissue slice, a tissue slice will also be a mono layer or few layers of cells, 1 or 2 layers of cell, that is a tissue slice with matrix, and a culture of cells will have just a single cells with some matrix. Okay. How can we model this? Now, you have understood things, right. Now, you know how to model things. So, you can always effectively put the effective impedance of the whole tissue as one resistor in parallel with a capacitor, and let us say the tissue is kept in a solution called PBS, PBS is the phosphate buffered saline. It is what is used to maintain the pH so that the cells do not die. So, that is again a salt solution. So, it has a resistance. So, RPBS, and these electrodes that are there below the cells, they offer a double layer capacitance at the interface between the, because there is an interface of metal and the lipid bilayer, right, of the cells. So, that is basically a double layer capacitance. That is why it is modeled like this. A double layer capacitance effective cell model. It is a very simplified model of the whole tissue. Okay, you can always improve on it, and the solution that it is in, this solution that it is in is the RPBS. So, this is, I just wanted to tell this to you, like you given a scenario, how will you model it in a very simple way. You can always make it more complicated. You can put a resistance in the path of the cell and tell like that it will be like this, not like that. So many things you can do depending on what kind of cell it is, and you can keep improving the model. This is one interesting example. Okay, this is how you model different types of cell organizations for different biomedical applications. This is a biomedical application, right, where we are trying to measure the monolayer of cells on an electrode and seeing, and if let us say we add some drug to kill the cells, how will the electrode measure the impedance, how will it change. This is how biomedical systems can be built from first principles, and the first principles we have covered now. All the first principles, frequency dependent behavior, DC behavior, what is a resistor, which part of the cell is a resistor, which part of the cell is a capacitor, all these things we have covered. Okay. So, I hope this gave you a very good introduction to basic electrical equivalent circuit modeling of cells and how they come together to form tissues. And in the next lectures we will see other interesting aspects of mathematical modeling of biological systems. Stay tuned for upcoming modules. And if you have any doubts, please feel free to post in the forum, and we will be more than happy to help you out to understand the concepts. Thank you, and see you in the next module.